All right, in this video, we'll be talking about development. We'll start at fertilization, and we will go through understanding how the zygote develops and how it can change patterns. All right, so fertilization basically is the idea when the sperm enters into an egg cell. One sperm will enter into the egg cell, the other sperm cells will be blocked. This is showing a progression, not multiple sperm cells, but you can still get the idea. There's two kinds of blocking that goes on. There's a fast block that goes on, which is basically an electrical potential charge or change to the membrane. Basically pulses back all the other sperm. Think about it that way. And there's a slow block, which is, you can kind of see it on this picture. So each one of these sperm kind of come equipped in order to kind of talk to this little dot on the egg cell. And so the slow block is basically where the surface of the membrane changes such that the sperm can no longer communicate with the egg cell. And so there's no way for them to have this interaction. So there's no way for another sperm cell to actually combine. Most animals have a variation of this process, and then the sperm nucleus will fuse with the egg nucleus and make what is called a diploid zygote. So here you kind of see this, the sperm and ovum are fusing in order to make that zygote. So we're talking about early development. This shows you, from fertilization, fertilized egg, the zygote, and then it kind of goes into the different stages of development, which we're getting ready to talk about. First thing to talk about is polarity. The fertilized egg will develop two poles, the animal pole and the vegetal pole. The animal pole is kind of the fast growing end and will turn out to be like the head, and the vegetal pole will be the rest, and it's a slower growing area, and it contains a lot of the cellular nourishment as the cell begins to divide. And then the cell will go from like a ball of cells to a hollowed out ball of cells. And this hollowed out ball has this cavity in the center called a blastule, and the blastule will end up forming with the body cavity of the organism. And some animals will kind of stop at this phase. This is kind of the end of their life cycle. They don't get a whole lot more complicated than this. And so other animals will continue to develop, and they have multiple tissue layers, which necessitates this process called gastrulation. Gastrulation is, think of it, so here's our hollow ball, and so you have this infolding of this ball to form this structure called a gastrula. And what you have then is you have an outer tissue layer and you have an inner tissue layer. And so I don't know that I have I do right here. Okay. And so you have the formation of, don't look at this one yet. You have the formation of the endoderm, which is the inside most. This is like going to be your intestines and a lot of your innards. And then you have the ectoderm, which is like, think skin. In more complex animals, this is going to be like your nervous system also is going to come from your ectoderm. And so for some, this is the end of the line. For some animals, they will stay diploblastic, which means two tissue layers, and these are their two tissue layers. This would be like cnidarians. I mean, if I just put some like some little tentacles on this thing, you would totally buy it. All right. But for other organisms, this tube that started will continue on through, and it will form what like a full digestive tract opening. I don't think I have a picture of that. I don't. Maybe back here I do. Okay. And so will form a full digestive tract opening. And so these animals will form a third tissue layer, which is called the mesoderm. And these animals are triploblastic, which means that they have three tissue layers, and they will form a mesoderm, as you can see. And the mesoderm is essentially where your muscles are going to come from, bones. The kidneys are part of the mesoderm as well. And so this forms like a third middle layer. Most animals fall into this category here, and then that leads us to the next discussion, which is of the body cavity, or the coelom, not coelom, coelom. All right, and so some animals are called acoelomate. You can see here they are triploblastic. They have all three tissue layers, but they do not have a coelom. They do not have a body cavity, which we'll talk about what that means in a second. And so they have to be really small. The body cavity, what that allows, so you can see a true coelom. What the body cavity allows is it allows for you to have space for your organs. If you bend over, you don't break in half, essentially. And so these little worms have to stay really small, these acoelomates. Next is the pseudocoelomate, and you can see they do have a body cavity, but the body cavity is not completely lined with mesoderm. See this red layer here is like the mesoderm, and this endoderm is not lined on its outside with that mesoderm, and so it forms what's called a pseudocoelom, which will be full of water and still kind of like a body cavity, but not quite. And then the last group are called coelomates. They have a true coelom, completely lined with mesoderm. So a little bit about pattern formation. And there are genes in the nucleus of all animals called the homeobox genes that form these patterns, and they control the expression of those patterns. And this may make sense a little bit more with this next slide. And so here, notice here is a mouse embryo, here's a fly, and notice they have similar genes. All right, they have similar genes in similar places, and you would expect similar patterns to develop. Obviously, this section of the fly is going to be different than that section of the mouse, but those Hox genes is what they're typically called, or the homeobox, is where these pattern genes are found, and they regulate and control how an organism develops. Different organisms can have similar Hox genes, like the fly and the mouse do, but obviously they're not the same 100% because flies and mice aren't the same thing. They have the degree of expression is different, and then you take an elephant. An elephant's just going to have more of these genes because there's a lot more elephant. And so anytime you have major changes in population or major changes like species changes, like macroevolution type changes, you can see these changes in the evolution of these Hox genes. And lastly, looking at some different body patterns here. There's three basic body patterns. The sponge has no symmetry. It's asymmetrical, which is a very primitive form. Only the sponge has that. The next one's going to be radial symmetry. And radial symmetry, think of a radius, think of a circle. I hesitate to say infinite lines of symmetry, but you get the idea. Like a sea star has 5 lines of symmetry, right? So it has a pentaradial symmetry. But most animals are going to fall into this category over here, which is bilateral symmetry, where you have a single line of symmetry that separates the left and right sides.